The overall goal of this protocol is to sporulate budding yeast efficiently in a 96-well format.
Although many large-scale screens have been carried out on the budding yeast, high-throughput studies looking at meiosis and sporulation have been limited. The technique described in this video has the advantage of sporulating yeast efficiently in a cost-effective manner in a 96-well format compatible with high-throughput screening.
I first got the idea for this method because I wanted to use a tiled 2-micron plasmid library in order to screen for high-copy suppressors of a mutant that could not form refractile spores. Initially, my sporulation efficiencies were poor in the 96-well format. I found this was due to inadequate aeration.
Begin this procedure by thawing the SK1 yeast strain onto a yeast extract peptone glycerol or YPG plate. Use a sterile stick to scrape a small amount of cells out of the freezer vial containing the glycerol stock and spread it onto a YPG plate. Grow the yeast for 12 to 16 hours at 30 degrees Celsius. Next, streak the yeast from the YPG plate onto a yeast extract peptone dextrose or YPD plate for single colonies. Grow the yeast on the YPD plate for 24 to 48 hours at 30 degrees Celsius. After 24 to 48 hours of growth on YPD solid media, start a 25 milliliter YPD culture using a single colony from the YPD plate. Grow this culture overnight in a 30 degrees Celsius shaking incubator at 220 RPM until the cells reach an OD600 of 4.0 to 7.